And so the key discovery was that Durrell and Twort found that they could propagate an entity which survived filtration and had the following properties. You could take what appeared to the eye to be a clear culture. It was not turbid. And if you took this sample and you looked at it under the light microscope, you saw nothing. There were no bacteria there. If you took this and you added some sterile broth and you incubated it, it didn't go turbid. Nothing grew, apparently, in the sense that it was understood that bacteria and other microbes could grow. And although not everybody believed them, they were convinced that there was something in there, even though you couldn't see it. And the reason why they believed that there was something in there was that when you added this to a culture of bacteria, you could see, they could see that the bacteria were killed. And that gave rise to the name of bacteriophage, an entity that eats bacteria. And there were all sorts of theories that were thrown out as to what might explain this behavior. But what Durrell in particular pioneered and developed was this so-called plaque assay, an incredibly useful assay which we still use today, and that includes all of you guys, where you can take this culture at this clear liquid, you can plate it out on a lawn in a Petri dish where these bacteria have been essentially immobilized in a thin layer of agar. And when you add this liquid to that lawn of bacteria, you can see areas of killing, referred to as plaques, that correspond to regions where the bacteria are killed and no longer growing. So you get a lawn of bacterial growth, and you get these plaques where the bacteria have been killed. And what was critical in this experiment was that Felix Durrell could show that he could take this clear solution, he could make dilutions of it and demonstrate that each of these plaques arose from a particle. It wasn't simply a chemical that you could dilute out and see activity or no activity. The activity was particulate in nature, such that each of these plaques arose from a single particle. And there were several reasons why that was very powerful. First of all, you could readily count how many particles were present in this culture simply by making dilutions, plating out, counting the number of plaques, multiplying by the dilution factor, and it tells you how many particles are present. And those particles, this is a test that we still do, and those particles we would then express in terms of a concentration of plaque-forming units, or PFU, per mil of the culture or the lysate. The second thing that was really important was the fact that even though each of these plaques arose from a single submicroscopic particle, if you repeat the assay and ask how many particles are present in that plaque, it's about 1 million, 10 to the 6, 10 to the 7, sometimes more, sometimes less, but it's a very large number. So what has happened is that this plaque was generated from a single particle. And even though they don't reproduce and grow and multiply by themselves, when they're present with their bacterial hosts, there is massive reproduction occurring. So you go from a single plaque to millions of plaques. And therefore, these were not simple chemical entities. These really were biological forms, even though it wasn't clear what they were, you couldn't see them. They had these very well-defined properties that confirmed that they were biological entities, and a great deal of characterization on their properties ensued over the next 30 years or so. It took the development of the electron microscope to give sufficiently good resolution to actually be able to see what these particles look like, and to take them from their somewhat abstract representational properties to being able to see them, to look at them, and to understand their morphologies, and to look at the different morphotypes. There's many, many different morphotypes of bacteriophages that have been described by looking at them in the electron microscope. But there's three that appear to be particularly predominant. And these are referred to as the myoviridae, the podoviridae, and the cyphoviridae. And the distinction is based upon the nature or the character of the tail. And so you can see this tail structure here. And in the case of the cyphoviridae, this is often a relatively long, flexible, and non-contractile tail. In the myoviridae, it is a contractile tail, and the best way to think about these structures is as a syringe, and that's probably actually how they work, so they are physically, can physically contract. And the podoviridae have short, stubby tails, podo as in foot. So they do have tails, but they're short and stubby, and you can just see this one here. So these are three of the most common morphotypes, and what we now know in thinking about these different types of bacteriophages is that each of these has a tail, but they also have a head, often referred to as a capsid, and you can see it here and here and here. The capsids vary in size, typically from 40 to 50 nanometers to maybe 100 nanometers in diameter. And the capsid is made of a protein shell, and it's that protein shell, that head, that contains the genetic material. It's believed that the vast majority of bacteriophages contain double-stranded DNA within these capsids, but clearly there are other bacteriophages that contain single-stranded DNA, and there are those that contain RNA as well. But these, I've pointed to these because these are three of the most common morphotypes. There's an organization, the International Committee for the Taxonomy of Viruses, not taxation, but taxonomy. We could tax them, I guess, right? And this this body attempts to classify and to organize the entire viral population. And I've just shown you this just as a flavor, really, of some of these classifications that show these other forms, enveloped phages, filamentous phages, and different forms of nucleic acid. Just to emphasize the fact that even though these three are the most common, there's actually quite a diversity of morphotypes and genetic types in terms of the genetic material.